There is a monitor shape. Yes, that's all right. Yes, now it's go. Yeah, please proceed. Thank you.
Okay. No, okay. Good evening, everyone. Thank you for attending my presentation. I am Siti Aminah Ibrahim, PhD student of Ehime University, Japan. My supervisor is Professor Dr. Koike Jima. We are working in Coastal Ecology and Conservation Laboratory located in Kochi University. Both Ehime and Kochi University are in Shikoku Island of Japan. My PhD study is investigating the intake of microplastic by estuary crabs, focusing on its translocation and the impact to the crab internal organs. Microplastic is present everywhere in our surrounding, including in our workplace, the laboratory, as contamination. This type of contamination is almost impossible to avoid. There are studies reported microplastic present in the laboratory background, such as from lab windows and lab coats. However, most studies were focusing on airborne contamination of microfibers due to the high number of microfibers reported. As at our knowledge, examination in terms of other form of microplastic and sources such as chemicals, equipment, and water were not yet performed. Microplastic were also found in the environmental and biota samples such as crabs, fish, and copepods, which usually taken back to the laboratory for analysis. These samples could have also been exposed to the microplastic contamination from the lab, and this could lead to overestimation of microplastic counts from the sample. It is important to evaluate the laboratory background before the analysis work took place. Procedure blank that used for background evaluation unable to solve the problem to measure samples with low level concentration. This is because small aquatic organisms are reported to be having microplastic with less than 1 to a few particles per individual. Being aware that microplastic is present almost everywhere and could interrupt the analysis, we initiated the preliminary examination on all materials and equipment that is going to be used throughout the environmental microplastic analysis work. Our objective of this study is to find the possible sources of microplastic contamination and find out the best cost-effective prevention measure. Here is the potential sources of microplastic contamination in the environmental sample analysis step. Microplastic in the lab is potentially to contaminate the glass filter, water, chemicals, and the lab space through airborne contamination. As from the steps, we could see that airborne contamination could happen at almost all main analysis steps shown in beige color because samples and equipment are exposed to the air at most of the time. Water contamination is the second high potential in interrupting the analysis work shown in blue color because it is used to rinse the equipment, which then used for dissecting, digesting, filtering, density separating, and staining of all samples. Samples are also exposed to the chemical microplastic contamination as shown in red, as the chemical needs to be added into the sample. The glass filter potential contamination, here colored with green, is used in all filtering work. In short, based on the environmental sample microplastic analysis steps, we then decided to examine on these four important potential sources of microplastic contamination and then find the cost-effective way to prevent it. Before we start to examine the potential source and in considering that microplastic could present anywhere in the lab, we applied the basic practice as to ensure that our focus is only paid to the contamination in the sources. The first step of our basic practice is by using glass and metal apparatus and reducing the use of plastics. The second one is all prepared solution is covered with aluminum foil at all times. Then all work were conducted in the clean bench installed with particle filter that cleaned with 90% ethanol prior to the work. At all times, cotton lab coat and sterile rubber gloves were worn and petri dishes were kept closed as much as possible. Generally, all microplastic were captured through filtering the solution using glass filters or left landed in the open glass filter. The same goes to our study, where we have done these general methods in examining all potential sources of microplastic contamination, as well as checking on the later part after the prevention measure taken. All samples obtained by the glass filters were stained with nyrate, drying in the oven for 2 hours at 50 to 60 degrees Celsius, and then observed under the microscope. This microscope is used in our study were installed with a Sony camera on top of the microscope to capture the microplastic image, and it is equipped with two optocored LED green lights allowing visualization of the fluorescence under the microscope observation. All sample is going through this process before detected and enumerated. Subsequent to the microplastic contamination found in all tested sources in the lab, we then take steps on applying prevention measure on every tested sources to find if the contamination could be reduced. Currently, microfiber filters are commonly using for microplastic detection, and we are using Whatman glass microfiber filters. It comes in many sizes and types, and in the study we tested GFC and GFF. Both glass filters were supplied at different price, where GFC is supplied 40 to 50% lower price compared to GFF. These glass filters were made from glass but supplied in plastic packaging with no specification such as plastic free. For glass filter contamination prevention measure tests, both GFC and GFF were pre-combusted in a standard furnace at 400 degrees Celsius for 2 hours. After its cooling down, they were gone through the same process described previously, then the glass filter observed. We found that pre-combusted glass filters was able to reduce the number of microplastic contamination in GFC and GFF at 40% reduction rate. Therefore, we decided to use heated GFC in all our tests due to its cheaper cost and equal filtering quality. The second common and important material used in microplastic detection in environmental sample is water. In our laboratory, the tap water is filtered through the ionized cylinder to get the ionized water and then stored in the plastic tank. So we tested all these three water sources. We used same procedures to test all sources of water. Water was collected from each sources after flow off for about 1 minute. We found microplastic in all water sources, and the ionized water had the lowest contamination. Hence, the prevention measure was applied to the ionized water. The ionized water was filtered with heated glass filter to get filtered deionized water, and to ensure the reduction of microplastic in the filtered deionized water, it was then refiltered with the second heated glass filter. We repeated this test for 10 times and didn't find any microplastic contamination in the second heated glass filter. Translated, the rate of reduction was at 100%. Earlier, out of three chemicals tested for contamination, the highest was found in ferrum sulfate and the lowest in zinc chloride. For contamination prevention measure in chemicals, all chemicals were filtered through heated glass filter. To test the effectiveness of the prevention method, the filtered pantone reagent and zinc chloride were refiltered using second heated glass filter. These were repeated 5 times for every chemical. We found that the reduction of the contamination was reduced at 100% in ferrum sulfate and zinc chloride, whereas in hydrogen peroxide was reduced by 71.43%. For airborne tests, generally, all the work for sample analysis is carried out inside the clean bench with particle filter. So we tested the effectiveness of the clean bench usage as well. For this, we place petri dish with damp heated glass filter inside and outside the clean bench while working on the samples for 30 minutes. The damp heated glass filter used in this study as it having high efficiency to capture the airborne contamination. The glass filter that placed outside the clean bench collected 17 microplastic per filter, from the airborne, whereas the glass filter placed inside the clean bench only have 5 microplastic per filter. This showed that clean bench installed with particle filter could be best tool to reduce the microplastic inside the clean bench with above 70% reduction rate. We had also done a preliminary evaluation on the prevention measure taken through all steps of environmental sample microplastic analysis. For this test, we took the prevention measure throughout the actual Krebs organ microplastic analysis, which are from the dissecting process to obtain the organ sample, followed by digesting and density separation using filtered Pantone reagent and zinc chloride, and filtering using heated glass filter (GFC). Then the glass filter observed under the microscope. While working on the whole step, we placed the blank, which is the damp heated glass filter, in open petri dish in the clean bench. Upon the completion of the analysis, number of microplastic found in Krebs organ samples was subtracted to the number of microplastic found in blank glass filter, on which equal to negative numbers considered zero. This finding showed our prevention measuring method successfully minimized the microplastic contamination in the lab through the biota analysis work. This prevention measure study provides a practical tool for the analysis work. Although the evaluation of contamination in the current study was only preliminary, it could offer solutions to improve the possibility in reaching accurate number of microplastic in environmental samples by monitoring and preventing the surrounding microplastic contamination. Bottom line, it is possible to do the simple prevention measure at low cost in order to minimize microplastic contamination in the laboratory and to achieve high accuracy in microplastic analysis work in environmental samples. Thank you for your attention. Questions are now welcome.
Thank you very much, Siti. Excellent presentation. We leave the questions to ask the participant to write to you, and write to us. We'll forward to you due to the delayed, program today. Is it all right with you? Yeah.
Yes, sure do.
Yeah, and we will also share your contact information to the audience as well, so to get further in touch with you. Yeah.
Sure.
Thank you once again. Appreciate. Yeah. Thank you, Dr.
Yeah,
Wang.
thank you so
Okay.
much.
By the way, Darren's program, there is one more presentation to be in this track. Due to the time, issues, we decided to move that program, that presentation, to, let me share the screen. Yeah, we move the presentation to tomorrow. Okay, I'm wrong file. Here I go. Yeah, okay. The presentation by Darren Ruby in Australia, characterization and identification of microplastics by quantum cascade laser-based rapid infrared chemical imaging. He will be presenting tomorrow. The new schedule will be announced very shortly on this. Okay. Thank you all for joining this session, an oral presentation. Once again, I appreciate speakers for joining this session, presenting it, and I regret the inconvenience caused with the little delay. Unfortunate. Okay. Thanks, Siti, once again. Appreciate it. Okay. And I'm ending this session here. So I'm moving all the participants to the day one panel discussion. We're also skipping the session break as well, because we need to catch up with the time.